As some of us had the pleasure of walking in her lab yesterday, I'm going to introduce Dr. Trudy Oliver, the professor of Pharmacology and Cancer Biology at Duke University. Let's give her a welcome.
Thank you, Dusty. Thank you, Lynn. Thank you, all of you, for being here today and sharing your stories. I have to say, your strength, your courage in the face of challenge, your humor at just the right points in time. It means so much to us. Getting to meet many of you yesterday and go through the lab was a really powerful and impactful experience. I've already received emails from people in my lab sort of saying, you know, this has been the most grounding and impactful, you know, moment in my career so far, and I'm going to cherish it for, you know, for a very long time. And so thank you for doing that as much as you can keep sharing your stories. You know, we really, really appreciate it. I don't, you know, when I give talks all around the world, sometimes there's not people like you in the audience. So sometimes we show pictures to try to, you know, make it a human story. But, you know, your presence and your faces and your smiles and your stories really go so far. And so I hope you can continue the work that you're doing. Is it possible to make the first slide show up? Wonderful. Okay, so I don't have to show this image to this audience, but as I said, when I go around and give talks, you know, we start with something like this to help people understand what the problem is, and so I'm sure that this, you know, triggers a lot of feelings for those of you in the room who have experienced this. I'm going to try to give a broad overview of research today, and I hope to make it as approachable to you as possible. Certainly, if you have questions or you want me to tell it in a different way, just, you know, raise your hand or jump in, and I'm happy to answer questions as we go. So when I was in the first 10 years of my research at University of Utah at the Huntsman Cancer Institute, this was a female patient who came in, and this is sort of an example of the lung tumor burden in her chest, and then this tumor burden in the rest of the sort of peritoneal cavity. You know, it had already spread to different organs, and she had a brain lesion, as you can see here. And, you know, many patients receive combination chemotherapy to start with, and you can have these profound, you know, responses where this is a lung tumor located here, and post-chemotherapy, you know, as far as the eye can see, the disease appears to be gone. But as this audience knows, one of the challenges is that, you know, tumors aren't always completely gone. There can be tumor cells hiding, residual tumor cells that, you know, can come back, and then we need additional rounds of therapy, and in many cases, we need new and different types of therapy to go after the tumors that rebound. And so this is what motivates us. How can we find better therapies to begin with, less toxic therapies? What do we do when the cancer comes back? What can we do to stop the cancer from coming back? Now, one of the early pioneers in beginning small cell research was Dr. Adi Gazdar, and he worked closely with a number of investigators, including another gentleman, John Minna, both of which later ended up at UT Southwestern in Texas. But what Adi Gazdar and John Minna were some of the first to do were to take patient tumor cells from lung cancer specimens and put them in a dish. And when they did that, when they put cancer cells in a dish to grow them, they noticed that they would grow in different patterns. And this was sort of the beginning of realizing that not every small cell lung cancer tumor is exactly like someone else's. So there's variation, or what we like to call heterogeneity, in the types of small cell cancer cells and tumors that grow. And they noted that they had these differences in the dish, but they also noted that if you inject these cells back into an animal, we use a lot of mouse models, if you inject those cells back into an animal, the tumors that would result in the animals also look differently underneath the microscope. So this was just further evidence that not all cancer cells behave the same way, even though we call them small cell lung cancer. Now, I don't expect you to interpret all of these graphs, but they're all basically to make the same point, which is after those initial studies in the 80s, many researchers came along and noted using different types of approaches, different methodologies, that yes, cancer, small cell lung cancer, they don't all behave the same way. So whether you're looking at what types of viruses can infect those cells, or what their methylation state is, which is just an indicator of what's going on inside the cancer cell, or whether you're looking at their sensitivity to cell death, how easily do they undergo death when you treat them with a stress, you can see that these are all different samples and different lanes. They're not all looking the same way. So there's lots of variation. And how do we understand this variation? And how do we ultimately identify therapies that are going to be, that each cancer is going to be vulnerable to? Now, just in the last, let's say, 10 to 20 years, there's been sort of a revolution in trying to understand the genomic identity or the genomic alterations that are common to cancer, because in other cancer types, non-small cell, for example, when we match therapies to the genomic alterations in a tumor, we can have great success. And so, of course, many wanted to apply that same concept to small cell lung cancer. And so studies were performed to identify common genetic alterations in small cell. And one of the things that was found that is important is that almost all small cell lung cancer tumors have alterations in two key tumor suppressor genes called RB and P53. So these are like the brakes to cell growth. And so what small cell often has are mutations that disable these brakes so that if someone steps on the gas pedal, there's no brake to stop the car. So they've sort of removed their key brake systems to growth. Now, it probably wouldn't be such a problem if nothing was stepping on the gas pedal, but there are alterations in genes that step on the gas pedal and promote growth and growth of the tumor. And those are called oncogenes, and some of the most common oncogenes that are altered in small cell is this family here, MYC, MYC-N, and MYC-L. So these three family members function a lot like each other, and small cell tends to use one of them per tumor to drive proliferation. And so the MYC family members are telling cells to grow, and at the same time, we're missing RB and P53 to step on the brakes. And so this is part of the genomic alterations that are driving tumorigenesis. Unfortunately, these are not genetic alterations that are easily druggable or easily fixable. And so investigators have had to be creative to look for other ways to create therapeutic vulnerabilities in the face of these alterations. And one of the ways we do that are try to make model systems that we can test our favorite therapeutic ideas and strategies in a more rapid and cost-effective manner. Of course, we don't want to experiment on people. We want to figure things out in the lab so that we can apply the things that work the best when patients come in. So one of the things investigators have done have been to engineer these alterations in animal models, and this has actually been very successful. Some of the early work showed that if you alter those two key tumor suppressor genes, RB and P53, specifically in the lungs of mice, the mice get small cell lung cancer that looks basically identical to the human disease. A pathologist wouldn't be able to tell that this was a mouse. And so these tumors look like small cell, they behave like small cell, they spread around the body to different organs in the same way that we see it happen in patients. And this is just an example of the variety that can happen in the mice. So this is two different examples that I showed you earlier of what human tumors can look like. We call this pattern where the cells look darker and more condensed classic small cell. And we call this sort of lighter shade of purple where the nuclei are a little bit bigger and more open. We call this variant small cell. And importantly, the mouse models can recapitulate all of that sort of what we call histological spectrum of tumorigenesis. And as I said, a pathologist really wouldn't be able to distinguish whether this is mouse or human. They're virtually identical. Now, just in the last 5 years, there have been another groundbreaking set of discoveries, which is realizing that heterogeneity in small cell behavior, why do they look different, why do they all behave a little different, comes down to the molecular level, a little bit more molecular and gene expression level than genetic alterations. I'll try to explain what I mean here. So these are over 100 human small cell tumors in rows or columns this way. And we can basically stratify small cell behavior based on expression of these 4 lineage-related, or let's say cell identity-related transcription factors. So these are just genes that are expressed in the cancer, and one of the most common of which is called ASCL1. So ASCL1 is highly expressed in this group of tumors here in yellow. And then we have 3 smaller groups of tumors that are defined by different factors. You can see they don't express as much ASCL1, so blue is low and yellow is high, but they do express either NeuroD or another factor called PAL2F3 or another factor called YAP. And this group is a little more controversial. That would be for a whole conference. We won't talk too much about that today. But just to say that these factors, you can think of them a little bit as biomarkers of different states of small cell. So we have this big group here and 3 smaller groups here. And when I talked about the oncogenic drivers earlier, this group that has high ASCL1 tends to have a lot of the MYC-L family member, whereas these 3 smaller groups of small cell that have low ASCL1 tend to have high MYC family expression. That will become important for some of the therapeutic things I want to say next. Now, importantly, each of these factors drives a different cell fate or identity, and I've kind of explained what those are up here. So cells that have high ASCL1 are very neuroendocrine, which is what you may hear small cell is. It's a neuroendocrine type of tumor. But there are these 3 other flavors where small cell takes on a little bit different identity. It can be more neuronal when the cells have NeuroD. It can be more what we call tuft or brush cell-like when cells have PAL2F3. And in the case of YAP, more mesenchymal or what we call inflamed. Now, this work that has happened in the last 5 years has really led us to start thinking about what are the therapeutic vulnerabilities that associate with these subtypes. And importantly, we and others have found that when cells are in these different states, they tend to have very different therapeutic vulnerabilities. So we think it matters what cell state a small cell is in. The models that I just talked about, the mouse models that recapitulate the human disease, can be used to figure out what genes are important in small cell. And since I just mentioned ASCL1, I wanted to mention a couple studies that have been done in animal models. ASCL1 is that factor that really drives the neuroendocrine state of small cell, and investigators have knocked it out of mouse lungs. And what you can see here is if you compare the normal animal model, these little purple regions are tumor here. So the mouse has a number of different lesions in the lung, and this is just a blown up magnification of these purple regions. When you knock out ASCL1, you completely get rid of small cell lung cancer in that model. So ASCL1 is really critical to the identity of those tumors. Those tumors don't express that other factor, NeuroD. So if you knock out NeuroD from that animal model, you have no effect on tumors. You can see they still have a number of purple lesions, not any different than this animal here. So that really shows that in some cases, ASCL1 is absolutely critical for the viability and growth and survival of these tumors. And that's an important finding that could be useful in terms of thinking about therapeutic capabilities. But now I want to say that context is kind of everything in small cells, so what applies to one group may not apply to another. So for example, if you take a different type of model of small cell, the RBP53 c-MYC-driven model of small cell, and you knock out ASCL1, you don't completely get rid of tumors, but you do shift their fate dramatically to other types of tumors. So the bottom line is that ASCL1 seems to be really critical for a tumor to be neuroendocrine. It can either, if you don't have ASCL1, you can hinder the growth of tumors, or you can shift their identity to something else. And we think that when cancers change their identity to something else, that their drug vulnerabilities are going to change too. And then another important finding related to signaling I just want to mention briefly is about the Notch or REST pathway. These signals or pathways have been shown to promote what we call the non-neuroendocrine or ASCL1 low type of cancer. So when small cell loses ASCL1, it tends to be more dependent on these pathways. And these are just shown here in some experiments in the lab where if you put Notch activation in cells that are growing like small cell, it changes their morphology. And you can see when you put Notch into cells looking at a Western blot, you lose the expression of ASCL1. So ASCL1 and Notch are sort of at war with each other, if you will, controlling what identity small cell is in. As we begin to understand what genes and pathways are driving the state of small cell, we made an important discovery in our lab in 2020, and that was really some of the first evidence that these states of small cell aren't just stuck in their state. So an ASCL1 tumor doesn't always have to be an ASCL1 tumor, and a NeuroD tumor doesn't always have to be a NeuroD tumor. They can change from one to the other. And so we used a number of sophisticated approaches, both in mouse models and in human cells, and showed that the ASCL1 type of small cell can become the NeuroD type and can ultimately become the YAP type. And that work prompted the work of many other studies, some of which are listed here, where others have taken different approaches than we did and basically found the same thing. And that is that small cell subtype can convert from one type to another, and it doesn't seem to go in just one direction. They can go back and forth, and that means what we call there's a lot of plasticity or ability to change. So you can sort of think of small cell a little bit as a shapeshifter. It can take on this form today and a different form tomorrow. And the reason why I'm telling you this, obviously this is a great challenge when we think about treatment, but we want to understand how they do it and to predict ahead of time which directions they like to go so that we can foresee that and incorporate these kinds of things into our therapies. So the study that I just showed you previously, we learned that one of the key factors to get from an ASCL1, let me just go back one, to get from an ASCL1 tumor to this YAP tumor, we found that MYC would drive this process and that it needed to turn on Notch to do it. And we found that Notch was critical for that process. So as we went and looked back at some of this human data, we realized that the cells that have high c-MYC tend to be what we call Notch wild type, meaning they're able to turn on Notch to get to these states, whereas these tumors here tend to have mutations in Notch, and so they can't turn on Notch, and they're more likely to be stuck in that state. And that brings an important point that genetics do seem to matter, because you might have a mutation in a tumor that helps lock it in a certain state, and that information might be valuable in terms of thinking about therapy. As I mentioned earlier, the fact that these states have different therapeutic vulnerabilities, we and others have taken a lot of different approaches to find therapeutic vulnerabilities in specific subtypes of small cell. This is an example of a drug study where we took different cell lines that are in different cell states and applied a large drug screening approach. And this is just an example of some drugs that worked really well. Those are the drugs in red. Specifically in this class of tumors in green. So we're sort of matching therapies to specific groups of small cell, and that study pointed us to a class of drugs called aurora kinase inhibitors. And we found specifically that tumor cells that have high c-MYC, as opposed to the L-MYC or the N-MYC family member, tend to be vulnerable to aurora kinase inhibition. And we proved that in different ways by activating different MYC family members in small cell lung cancer cells and showing that only those where we activated c-MYC had tremendous sensitivity to this aurora A kinase inhibitor called alisertib. So cells that had N-MYC or L-MYC or no MYC didn't really respond to the drug, but those that had high c-MYC responded very well. And so those are the kinds of examples we're using to find drugs to test in clinical trials. And that drug in particular was tested in a clinical trial. This is by Tafik Awanikoko's group, where they had about 80-something patients. And these patients, after they had failed chemotherapy, received the aurora A kinase inhibitor with Taxol. And if you were to just group all the patients together, it would look like there was no effect from the drugs. But if you went back into the patient group and you said which tumors were c-MYC positive versus negative, now you see very striking differences in the data, which is that those patients that had high c-MYC in their tumors that got alisertib instead of a placebo, they had a significant increase in survival compared to those that got Taxol alone. And then the opposite was true if the tumors were c-MYC negative. So this was one of the first indications from results in the lab that were translating to exactly what we would predict in a patient tumor. So those are the kinds of things that we want to use discovery-based approaches in the lab to find drugs that will work and work in the right subset of patients in the clinic. Other ways in which we found small cell to be heterogeneous between patient to patient are using human cell lines in mouse tumors and looking at metabolites, or what we call metabolic profiling. Cancers grow a lot, and so they need a lot of nutrients, and they do a lot of, they need a lot of building blocks in order to double their mass and make more. And we can take advantage of those metabolic vulnerabilities by depriving them of metabolites that they need to grow. And these studies were one of those that helped us find a specific therapy called arginine deprivation. This is an amino acid that you often get from your diet, and it can be drugged with this pegylated form of an enzyme called arginine deaminase. Most of the cells in your body can make their own arginine, but sometimes tumors can't make their own arginine, so they become very dependent on getting it from the body. And this enzyme chews it up and deprives the body of arginine. And we found that specifically in MYC high tumors, but not MYC low tumors, that this drug can really promote survival much farther beyond the standard of care. So untreated animals are in black. Animals that received combination chemotherapy doubled in their survival. And then those that got the ADI-PEG20 compound saw nearly a 3 or 4-fold increase in survival compared to the standard of care. And it's actually a relatively very safe drug. There are very few side effects. And so our group would really like to see this drug, particularly in combination with something else that could further delay resistance, go into the right patient population, particularly those that have tumors that are high for c-MYC. And those are studies that are ongoing. As I mentioned earlier, we've also used different approaches to see that tumors are sensitive to different types of cell death. This is not our work, but some of our work shown earlier with BH3 profiling. This is an assay looking at a type of cell death called ferroptosis. You can see that there's some cells that don't respond at all to ferroptosis inducers, whereas others are quite sensitive. And so we think that subtype can and does matter. This is sort of a pie chart just sort of showing that, very brief summary, and I wouldn't treat this like the Holy Grail, but sort of as an example that we found that different groups of small cell have different therapeutic vulnerabilities more so than others. So some of those that have already been talked about, DLL3 inhibitors and CES6 inhibitors, we predict are going to work much better for the ASCL1 group of small cell, which is of course the majority. But we need to anticipate that if cancer cells move to these other states, they may downregulate some of these targets, and we may need to then employ therapeutic strategies that are more vulnerable to these groups of small cell. So just in the last really 10, 12 years since I've been working on small cell, when I started the lab, we were really thinking small cell is just a single disease. And then within about 5 years, we were sort of thinking, oh my goodness, small cell is really a multiple subtypes, and we need to be subtyping patients to think about strategy, until our more recent studies realizing that they can change from one to the other. And so maybe small cell is one disease, but it's one very plastic disease that has the ability to shapeshift. And so we need to understand how cancer cells do this so we can ultimately stop it. And that sort of brings me, in the last couple slides, to talk a little bit about immunotherapy, because as this group knows, this has now been brought into first-line treatment for small cell, and it does improve survival. But I would say we know that it's not enough for most patients, and it's not enough for enough of patients. And so what can we do to better understand the immune system interaction with small cells so that we can get the body involved in fighting the cancer? Because when we apply most of our therapies, those therapies are static. They don't change. They just go after one target. But if the cancer can change and we're going after it with something that doesn't change, we're sort of behind the game. So if you think about it, the most evolvable type of therapy we can use at our disposal is the immune system. That's what the immune system is designed to do, is to be able to evolve and respond to things in real time. And so, of course, we would love to get the immune system involved and teach it to go after the cancer. So investigators are thinking about that. We call it, mechanisms to warm up small cell, because small cell is thought of as a cold immune tumor, meaning that it's not talking to the immune system very well. It's not presenting the things about it that make it foreign. And so it's almost a bit invisible to the immune system. So how do we get the cancer to sort of show on its surface that it's doing something wrong, and get the immune cells to come into the tumor and recognize it and want to attack it? So different people are taking different strategies to find some of the signals that are associated with good immunotherapy response, some of which in this study are listed here: higher MYC, higher rest, higher activation of Notch signaling. All of those factors have been associated with the more ASCL1 low group of small cell. So investigators are trying to figure out, both what correlates with good immunotherapy response and how can we promote those things to make for better immunotherapy response. And I don't think you can probably see this one too well, but this is just to say lots of different labs are going at this, looking at everything from epigenetic drugs to cell cycle drugs to DNA damage repair related drugs, and then of course things on the cell surface that can talk to the immune system. And I'm sure you'll hear from many of the investigators here today about some of the exciting things in that space. And so overall, just to end on this note, one of the major focuses of our lab is to really understand these mechanisms of plasticity so that hopefully we can ultimately inhibit it, because our work really suggests that small cell acts a bit like a moving therapeutic target. And we really want to control this landscape. So our work seeks to understand these, barriers to how do we, when we find a cancer cell in a certain state, how can we erect barriers to keep it from moving or changing? And because in many cases, when a patient comes into the clinic, they have a tumor that has cells already in different states that are responding to therapies differently, are there things that we can do to make these cancer cells behave more uniformly, to behave more like each other and consistently, so that when we apply a therapy, we're more likely to have a positive and long-lasting outcome. And for those types of studies, we really need strong computational computer support, everything from taking advantage of AI-based, computer algorithms to systems network modeling. We borrow technologies from different fields to ultimately computationally try to predict how the cancer cells can move and what their vulnerabilities are going to be when they move to these different states. And with that, of course, I want to thank the people in the lab that do the work, because I don't do the work. I just get to travel around and meet people and talk about the work. Many of you got to meet some of our lab members yesterday. This was a retreat that we took to Asheville, North Carolina, to just be with each other constantly and talk science and bond and, get our passion going to come back and do important work. And it was a really, really valuable experience, and I really appreciate you guys for talking to our lab members yesterday. Like I said, you were so incredibly inspiring to them, and I know that you've motivated them to go into the lab and work harder and faster. Of course, we couldn't do anything without gobs of money. It takes a lot of money to do the type of work that we do, both for the people and the expensive technologies and the animal models. And, I don't think our lab is the hugest lab, but with 12 to 16 people, we spend about $1 million a year. So it takes a lot of funds to keep things going, and we have to reach out to lots of different organizations. And this is a snapshot of some of those that have funded our work. And I also want to say, you may have appreciated from your meetings now that it really takes an international village. We have collaborations with groups in Germany and the UK and of course with pharma. We work with pharma to test drugs in our animal models. And I really think it takes all of us, including you, telling your stories. And so thank you very much for letting me participate with this group. And I'm happy to take any questions about science. And thank you for your attention.
Questions from anyone? Maida?
Hi, Dr. Oliver. It was a pleasure meeting you and your troops yesterday. And I did ask you a question about biomarkers and why you can't use blood biomarkers to detect small cell lung cancer. And you said, well, not yet. But is there a reason why not yet?
Well, I would say people are working on that actively. I think Dr. Lovely can probably talk a little bit about that today. But, we can easily and safely take samples from the blood, and different investigators are working on, which type of analyte to look at from the blood. So there's circulating free DNA that some are using that can sort of, they hold marks of whether they're in the ASCL1, NeuroD, PALTOF3, and YAP state. So some people are looking at that. There's also sometimes circulating tumor cells, intact tumor cells in the bloodstream. Others are using that as a source of material. And there's probably others as well. So that's a really heavy investigation of research. I think it just hasn't quite totally made it to the standard of care in the clinic. But I would imagine that in a short amount of time, we'll start to see things like that correlating with, are you a good candidate for a clinical trial such as the DLL3 or the SEZ6? That's my hope, and I think Christine probably knows more about that, so I'll let you ask that.
Thank you, Dr. Oliver. Any other questions? Okay.
First off, I want to thank you for the information, and knowing that the enemy is in multiple variations helps visualize what we're praying for. Also, is anybody studying old smokers that have never gotten cancer?
I'm sure there are, and certainly I've heard about sort of population science studies looking for like genetic variants that might be, protective in those that, smoke for a really long time and don't have cancer. So I think that's something else to add that maybe hasn't been represented yet so far in my data of just like the sheer number of people in population health that are doing these kind of, whole genome studies. It's a little bit out of my wheelhouse, so I can't comment too much about, what is known. The opposite direction we know more about, which are like genetic variants that are more predisposing to cancer, but in terms of like protection amidst, heavy smoking, I'm not sure.
I have a question I'd like to ask Esther. It's kind of a multiple question, but on these subtypes of small cell lung cancer, it's, from what I understand, it's not necessarily the standard of care for when they do the biopsy to do the subtyping, but is there something that we can do to, as patients, to ask for that? And then the other thing is, if you have a certain subtype, is that an indicator of, how well you might do or not? Is there any indicators about if you're, YAP or whatever, then that you have your chances for survival might be better or worse than another subtype?
Yeah, there are some subtle differences by subtype. I don't know if you can see this study here, and I think there's been some more recent ones than this one actually, but in this case, survival, and these patients received etoposide, platinum, and I think atezo, if you can see that better in the back of the room. Is that what that says right there? But this is small cell ASCL1, NeuroD, PALTOF3, and what they called the inflamed group. And in this case, the patients that had the lack of ASCL1, NeuroD, and PALTOF3 in blue, so I like to call those lineage negative or triple negative, those did better when immunotherapy was included in the treatment. And among all of these, the PALTOF3 group I think regardless of immunotherapy status, tends to be a little bit worse in terms of overall survival. And that's consistent with that group tending to have high c-MYC, which is a really powerful growth driver. And in terms of your first question, I know big groups like Sloan Kettering and probably MD Anderson and others are working on sort of CLIA certified, stains of biopsies and other ways of looking at biopsies for subtype. They're probably not going to make it part of routine until it has a therapeutic implication, So right now, it's like it means a lot to researchers, but it doesn't necessarily yet mean a lot in the clinic about what are we going to do for you in terms of standard of care. So obviously my group, with all the findings we've had with certain therapeutic vulnerabilities working particularly well in the MYC high groups, which might be applicable to a PAL2F3 positive patient's tumor, or if you find high c-MYC in a biopsy, we're working hard to get some of those vulnerabilities in the clinic, and we need to be able to run trials and show that it's effective specifically in patient tumors that are high for that to begin with, right? So there's a little bit more work to be done before I think it's going to immediately translate to therapeutic decisions.
Thank you. Yes.
Just with the awakening of machine language and AI, when you look to the future, is there like one thing that you're kind of thinking, oh wow, that would be so awesome if we could do whatever with the data, with whether it's organoids or whatever? What runs through your head?
Yeah, it's a great question. I think I've just barely explored ChatGPT and seen sort of like the most baby glimpse of the power of sort of AI. So I think my mind is, I can't quite imagine yet what we can do. But I can imagine that we have the power to totally understand the trajectories that we've been studying. Like small cell, really follows these conserved developmental patterns that go back to sort of embryonic development and development of the lung. And actually what happens when our normal lung sustains an injury and needs to repair itself. So it follows these developmental signals where the stem cell-like cells in the lung need to proliferate and make more of themselves, and then go differentiate, as we call it, into the different cell types. Our work suggests that small cell really follows those patterns and that they're predictable. And so, using AI, you could theoretically understand every aspect about how a cell chooses these different fates and what ways and directions they're going to go. And as we understand that, we should very much understand the signals and the pathways that if we were to inhibit could restrict this plasticity and ideally funnel cells into one state, a state that we want them to be in for our best therapy. And I think, as I mentioned earlier, I think if we don't get the immune system involved, this is, we're going to be like a dog chasing its tail, forever and ever, looking for the next thing. So my hope would be that we would use that information to find the most immunologically stimulating state to sort of educate our immune system to go after the tumor. And, but I need someone brighter in the AI field to, help us put that information together to get the immune system involved. Yeah.
Hi, hi Trudy. Just wanted to make a quick comment. So with my clinician hat on. So first of all, Trudy is amazing and her lab does amazing work. Yes. We'll probably talk a lot about biomarkers throughout the course of this conference, and biomarkers are fantastic. And I'm going to speak a minute about the other type of lung cancer, which is non-small cell lung cancer. And we have a ton of biomarkers for non-small cell lung cancer that we test for every single day. In fact, it is standard of care for non-small cell lung cancer that you must get biomarker testing. And if you don't, you're mistreating your patient. However, for small cell lung cancer, there is no biomarkers that we standardly have to get for small cell. And so there are 180 degrees apart right now. And so herein lies one of the conundrums that we deal with in the clinic, right? So we love biomarkers. We talk about biomarkers. We want biomarkers. That helps us bring precision medicine or personalized medicine for our patients, right? We want to tailor our therapies to the patients. We want to bring research to our patients. How do we do that? How do we bring amazing research from the laboratory to patients in the clinic? This is where the translational component comes in, right? How do we bring all this amazing research from Trudy's lab, from Treparna's lab, from Simon's lab, to the patients that I see in my clinic, from my own lab, right? That's where you guys come in. That's where the clinical trials come in. Because we need to bring this research to patients, and the only way that happens is if we're able to take these findings and actually put it in patients, that we have patients participating in clinical studies, that we actually reverse the translation, that we say, hey, we have patients on clinical trials, we study those samples, we bring it back to the lab. It has to go both ways. It has to go from the lab to the clinic, from the clinic to the lab. Otherwise, it doesn't work. And I will tell you, because I walk in both of these worlds, in the clinic and in the lab, it is very difficult. For many reasons, it is difficult to enroll patients with small cell lung cancer in clinical trials. We will talk about that, I'm sure. It is difficult to get tissue samples from patients with small cell lung cancer for many reasons that we could spend an hour talking about. But if we want biomarkers for patients with small cell lung cancer, if we want findings from Trudy's lab and other labs to come into the clinic, we need you all. And your voice is so incredibly important. But it is important you hear this. There are no biomarkers standard right now on April 16th, I think that's today's date, 2024, for small cell lung cancer. But that's what we need. Thank you.
Thank you. Okay.
All right, thank
you
you
all so much.
Thank you, Dr. Oliver. Oh, sorry, you have one more question, Dr. Oliver.
And I'm going to show my ignorance. But I love research guys. They're awesome. They give their life to the lab. But does the body get resistant to the therapies, or does the small cell get resistant to the therapies?
It's actually a great question. I would say the majority of the time it's the cancer becoming resistant to the therapy and not the other way around. However, there are some therapies, and I would use the arginine drug that I mentioned as an example. But the arginine deprivation drug is not yet approved for small cell treatment. That's one of the things that we're working on and would like to see. But the body can develop antibodies to that class of drugs, and in that sense sort of disable the efficacy of it. So there are things that the body can do that can be inhibitory to some therapies. But the vast majority of the cases, it's just the cancer evolving to a state that no longer responds to that therapy.
Right. So it shape shifts into something else, and then you have to use other therapies to try to control it.
Exactly. That's it. Thank you.
I think your wife has a question.
Do y'all ask for small cell lung cancers to give their tissues or their whatever? Because I've told my doctor several times I've been willing to do whatever they need for to take from me to make it better and look for more research. How does that work? And how do you get signed up to do that if you're to your doctors and they don't do nothing?
Yeah, that's also a good question for Christine, because what I have seen just in my researcher point is when we're at an academic medical center, and I've been fortunate to be at places like that, like Duke or University of Utah, I, as a researcher, can work directly with a clinician and a surgical team, and we can set up, consent procedures to ask a patient for these things and receive them. And for example, when I was at receive blood from patients and use that to isolate material to look for biomarkers and to inject circulating tumor cells back into mice and make basically a model of that patient's tumor in an animal. And that was very effective there. But I think some patients are probably treated, not at academic medical centers, and they may have no protocols in place to sort of ask for this material. They may not have collaborators to make use of it. So at least in my experience as a researcher, I like when patients are at some big academic medical center where they are interested in doing research and they do have these things in place. And maybe Christine can speak to the other half of like, you know, what does that look like from a clinician perspective?
Sure. Yeah. Yeah, so Trudy and I are very good friends, so we're going to refer to each other by our first name. So, which is another wonderful thing about the lung cancer community. So you all are good friends, and we all are very good friends as well. So we're going to refer to each other by our first names. So your question is a really good one. And the truth is, you know, studying mice is great, but studying people is much better. And so, and we learn from both, and we need to learn from both. And so at big medical centers, most universities have tissue collection protocols where if you're going for a biopsy and we need one core, which is like a little hollow piece of tumor that you get for a biopsy, sometimes you'll get 2 cores, and the extra core goes for a research collection. If the protocol exists and if there's money to support it, which actually the latter, not trivial. That has to all be done with philanthropic funds because, you know, that has to be, somebody has to support the money to even just bank the tissue to keep it in a refrigerator. So there's a lot of logistics behind even collecting tissue when a biopsy is being done as part of standard of care. However, and here's the important point, only about 5% maximum of patients in our country get their care at universities or major academic medical centers. So that means only the minority of cancer patients in our country are actually getting care at like big universities. And I work at a big university, and I've always worked at a big university. I'm not saying it's better. We have a tissue collection protocol, but if we're going to democratize cancer care, then we need to make sure that we are studying everybody, not just those patients who are getting their care at big cancer centers. Because there are many amazing, wonderful private practice or big oncology groups that also have outstanding cancer care, that also have really access to great clinical trials as well. And so we have to meet patients where they're at. This is all to say, collecting tissue is not trivial. There are many big centers that do it well. It's not easy to just go to your doctor and say, hey, I want to donate tissue. How do we get around that? One thing is liquid biopsy, which Simon will talk about as well because he's an expert in this. We have done studies in liquid biopsy in my lab as well. Thank you for the shout out, Trudy. And I'm going to start actually a direct-to-patient liquid biopsy protocol through a lung cancer organization. So you can actually donate blood if you're a patient with small cell lung cancer. That is still evolving, so hopefully we'll launch sometime later this year. That is one mechanism, but we need much, much bigger studies like that, and it's very expensive. But that's a mechanism for research, to bring research to patients instead of having to only get your care at a major academic medical center. So I think liquid biopsy will democratize a lot of access to care, where, you know, getting tissue is just not possible at a lot of community, what we call community-based cancer centers.
Thank you. Oh, you have another question.
I just have a quick question. What would they typically do with the tumor they took from me for my biopsy?
Yeah. So,
And I'm not in an academic, I'm at a community-based cancer care center.
yeah. This is on. Great question. So first, I by no means want to say that one is better than the other, because there are some fantastic university centers, there are some fantastic community private practices. You know, I think every patient has to be with a doctor and a team that you feel most comfortable with. And I always tell my patients that you'll meet many doctors in your life, and just like you meet many people in your life, and some of them you're going to love and some of them, meh. So you have to find a doctor that you really, really connect with. That is incredibly important because that doctor is your quarterback and you have to trust them 1,000%. So wherever you get your care, loving your doctor and trusting them 1,000% is so important. So what happens when you get a biopsy? Great question. So you get a biopsy, it gets put in this little tiny block that we call FFPE for formalin fixed, paraffin embedded. That's just the fancy word for your tissue lives in like a semi-solid glue that gets put in a warehouse somewhere. And it's kept for medical legal purposes for many years. And it's important because at some point in time, if you are on a clinical trial and they're like, hey, we want to test your biopsy sample, they're going to pull that sample and say, hey, we're going to pull your biopsy from a year ago or from 6 months ago, and we're going to use that for your clinical trial enrollment. And so when we use tissue from patients, we use it carefully as well because we never want to potentially sacrifice, hey, do you need a clinical trial down the line? And we don't want to misuse tissue. So we call it tissue stewardship. Tissue is gold, especially small cell lung cancer tissue, because there's not much of it. The biopsies are often very small. There's a lot of dying tissue on the biopsies, which is ironic, right? You're like, oh, this is such an aggressive tissue. But the truth is, it dies actually pretty quickly, even before treatment. And so we don't have a lot of material to work with. And that's been one of the reasons that small cell hasn't advanced as much as some other cancers have, because of limited access to tissue. So your biopsy is living in a warehouse somewhere, probably. When you get the biopsy, they cut what's called slides, where they use the slides to look under the microscope. Those also live in a warehouse.
And Dr. Lovely is going to speak later if you have a question specifically for Dr. Lovely.
I was just curious, out of all these various research labs, have anybody attempted to establish a centralized tumor bank, tissue bank, where the tissue samples can be sent and then researchers can draw from that?
Yeah, another great question. And yes, so later I'm going to do something different. I'm going to do a fireside chat where instead of like telling you about my research specifically, which, by the way, I'm a researcher too, so I love to talk about my research, but we're going to do just, I think, full 45 minutes of just Q&A. So you can ask me anything you want, clinically, research-wise. There's no such thing as a dumb question. We'll take them all. So a centralized repository. I'm going to give you an honest answer to that. The honest answer is it's challenging because different centers have different rules on sharing tissue because number one, it involves patient safety and patient privacy. And so, yes, we can share tissue. And there is something called the U24 Coordinating Center for Small Cell, which Trudy has been one of the leaders of, so maybe she can comment on that. And so I think there is resource sharing of tissue back and forth between centers and even internationally for small cell. But you can't, for example, as a patient, just say, hey, I want to send my tissue somewhere yet. Hopefully one day some of the barriers will be gone. Trudy, do you want to comment on the U24?
So when the NCI or NIH designated small cell as recalcitrant cancer, and I think it was 2012, they set up a lot of funds to make this coordinating center, which has been led historically by Charles Rudin at Sloan Kettering in collaboration with John Minna from Texas, and did more recently Lauren Byers from MD Anderson and myself. And so I'm a little bit more on the models side of the consortium. But it has led to some cooperation in terms of samples being sent to Sloan Kettering for certain types of genomic analysis and put into a repository called CBioPortal that everybody now has access to. But it's still limited. We would like to have thousands of samples. This is hundreds of samples. And there still are lots of challenges, as Christine said, in terms of getting different organizations to sort of cooperate, and the sheer magnitude of sort of paperwork related to, sharing private health information, can be a barrier that takes a lot of time and paperwork, as I said. So anyway, those efforts are ongoing. I hope to see things get better in that regard, and that we get large databases that are sort of publicly available to all researchers to take advantage of.